Today we can discuss about acid base homeostasis or acid base balance. So acids that can donate the H plus ions when in solution, that is in solution these acids get dissociates to release H plus ions. And there are some substances having H plus ions but that cannot be released in solution means, or that cannot be dissociated to release H plus means they are not acids. Such hydrogen ions when tightly bound to the molecular structure of that compound. So such substances that cannot release its H plus ions or protons in a solution is not considered as acid. So acids are the substances that can donate the H plus ions or proton. So coming to bases that will accept the proton or accept the H plus ions. So simply imagine hydrochloric acid. We all know hydrochloric acid is an acid. And see what happens in solution. In solution, this hydrochloric acid dissociate to release the H plus. So acid that will release the proton or release the H plus ions. And coming to one base, for example, bicarbonate ions, HCO3 minus. HCO3 minus will accept H plus ions. So it's a proton acceptor and form H2CO3, carbonic acid, one weak acid. So come to this HCl. This HCl, it is a strong acid, we know. This acid dissociate or ionize completely in solution. So this HCl lose its proton and form a conjugate base. Chloride ion is a conjugate base. See here also carbonic acid. Carbonic acid also lose its proton and produce a conjugate base. Base, we know this bicarbonate is a base. So strong base it is. So conjugate base. And since this HCl is a strong acid, that will produce a weak conjugate base, chloride ion. Whereas coming to this carbonic acid, it is a weak acid, we know, because it dissociates or ionize partially. So this weak acid loses proton and produces strong conjugate base. So strong acids produce weak conjugate base and weak acids produce strong conjugate base, or weak acids dissociate to form a strong conjugate base. So that's about the conjugate bases. Now coming to amphoteric substances. There are some substances that will behave both as an acid and as a base in a solution. For example, water, again ammonium. So now we can deal with the physiologically important acids and bases. We know physiologically important acids include phosphoric acid, carbonic acid, lactic acid, pyruvic acid, etc. And these acids are dissolved in body fluids. Coming to physiologically important bases that include bicarbonate, HCO3 minus, biphosphate, HPO4 2 minus. All these are the important physiologically important bases. Henderson Hasselbalch equation. We know pH is equal to pKa plus logarithm of concentration of a base to the molar concentration of acid. So that's about the Henderson Hasselbalch equation that will give a relation between the pH, pKa of a weak acid, again conjugate base and the concentration of acid. So that's about the Henderson Hasselbalch equation. Now see, when this acid is half neutralized means, when this acid is half neutralized, this pH will be equal to pKa. Let us see. pKa plus, if it is half neutralized means this will be 1. So log 1. So that results in pKa. So that pH will be equal to pKa. So we can give a definition for this pKa. pKa is the pH at which the acid is half neutralized. So uses of Henderson Hasselbalch equation, we can find out the pKa. When pH and the concentration of base to acid is given, we can write here salt also. Concentration of salt by concentration of acid also. So we can find out the pKa if pH value and the concentration of the salt or base to the acid is given. And also we can find out the concentration of the base to acid, concentration of the salt to acid if pH and pK values are given. And again we can find out the acid base status of a patient by knowing this Henderson Hasselbalch equation. So these are the important uses of the Henderson Hasselbalch equation. Now we can move to the buffers. Buffers, we know buffers are the substances that will resist the change in the pH of a solution when an acid or alkali is added. We know the pH of a solution changes when extra acid or extra alkali is added to that solution. But these buffers are such solutions which will resist. That means that cannot change, or that will not change the pH even if we add acid or base, no problem. So buffers are the substances that will resist the change in pH even if acid or base is added to that solution. So there are two types of buffers. There is either a mixture of weak acid with a salt with a strong base. So weak acid, salt of the strong conjugate base, or mixture of a weak base and salt with the strong acid. So weak base plus salt of the strong acid. So we can categorize these buffers into two types, that is either a mixture of weak acid and a salt of strong conjugate base, or weak base and a salt of the strong conjugate acid. For example, we can move to the bicarbonate buffer. We know bicarbonate buffer is having carbonic acid and sodium bicarbonate. It is a mixture of carbonic acid and sodium bicarbonate. Carbonic acid, we know it is a weak acid, and bicarbonate is a strong conjugate base, and the sodium bicarbonate means sodium salt of the strong base. And so many other examples. For example, acetate buffer is having a mixture of acetic acid and sodium acetate. Again, phosphate buffer is there. Now we can discuss about this buffer capacity. So buffer capacity is the ability of the buffer to resist the change in pH when acid or alkali is added. And the most effective buffer range means that is even if the concentration of the base to the acid is 1. So now we can move to directly this acid base balance. We know the pH of plasma, that is 7.4, and the range we can give, that is 7.35 to 7.45. If the pH is less than 7.35 means low pH means that is acidity. No, so acidosis, and this acidosis we can see CNS depression, coma, etc. Coming to this alkalosis, if the pH is greater than 7.45. If pH value exceeds 7.45, that results in alkalosis. Alkalosis means pH is above 7.45, and that leads to neuromuscular hyperexcitability, again tetany, etc. And life is threatened if this pH value is below 7.25 or if it goes above 7.55. So this pH maintenance, and the range of pH maintaining is very important. So there comes the importance of these buffers and all. And our body have a mechanism to regulate this pH in normal range. There are three mechanisms we can see, that is by the buffer system, the buffer of the body fluids, or by the respiratory system, or by the elimination or excretion by the kidney, that is renal mechanism. So by these three main mechanisms, our body can regulate the pH or maintain the acid base balance, or we can maintain our pH at 7.45, or this range 7.35 to 7.45, that range can be attained. During metabolism, we know so many acids are produced in our body. For example, keto acids, lactic acid, pyruvic acid, phosphoric acid, sulfuric acid, so and so. So these acids are produced in our body as a normal metabolism. And if these acids goes on producing like this and there is no mechanism for this one eliminating this acid, means what happens? There will be change in the pH. So definitely our body has the mechanism to eliminate these acid produced. And we can categorize these acids as volatile acid and fixed acids. Volatile acid example is carbonic acid. Carbonic acid is the best example for the volatile acid that is produced in our body by normal mechanism. Fixed acid, the other non-volatile acids that includes in the fixed acids, for example, keto acid, pyruvic acid, lactic acid, phosphoric acid, etc. And this volatile acid that can be eliminated by lungs as carbon dioxide. The other fixed acid that has to be buffered first, and the H plus O formed is eliminated by the kidney. So there comes the importance of these three mechanisms for regulating the acid base balance, that is the buffers of the body fluids, second one the respiratory system, and third one comes the renal mechanism. So these three mechanisms are important, very very important for the maintenance of this acid base balance. So bicarbonate buffer, we know two constituents, or it's a mixture of weak acid and the salt of the strong base, that is carbonic acid and sodium bicarbonate. And here this coming to this bicarbonate buffer, it is the most significant buffer in the body. Almost 65% of the buffering capacity in plasma and 40% of the buffering action in the whole body. That credit goes to this bicarbonate buffer. So definitely this bicarbonate buffer is very very important. And see, when we move to this its components, that is bicarbonate and carbonic acid, we know bicarbonate, it is the metabolic component, and that is regulated by the kidney. And carbonic acid, it is the respiratory component, that is regulated by the lungs, by respiration. And see, we move to this Henderson Hasselbalch equation. We know pH is equal to pKa plus log salt by acid, or log concentration of base by concentration of acid. So the square bracket that determines the molar concentration. So molar concentration of the bicarbonate to carbonic acid. So we can find out at which ratio of this salt to acid this pH will be regulated to 7.4. So we can find out by applying the values to this Henderson Hasselbalch equation. We know pKa of this acid is 6.1. And here the concentration of, molar concentration of bicarbonate in blood is 24, and that a carbonic acid, molar concentration of carbonic acid in the blood is 1.2. So applying this value, we will get log 24 by 1.2 means log 20. So 6.1 plus log 20, that is 6.1 plus 1.3. So definitely that value becomes 7.4. So the pH will be 7.4 if the concentration of bicarbonate to carbonic acid ratio, it is 20:1. Then when the concentration of this bicarbonate to carbonic acid ratio is 20:1, then this buffer shows the most effective bufferness, or buffering capacity will be high when this ratio is 20:1. So we have seen the ratio of bicarbonate to carbonic acid is 20 in order to become the pH 7.4 under normal condition, and this is actually greater than the theoretical value 1 that you have studied. So why? Because here the bicarbonate is said to be alkali reserve, and its value must be sufficiently high to meet the acid load produced in our body by normal metabolism. So actually during this metabolism, the alkali produced is very negligible, but the acid load is produced. We have seen that. So in order to buffer all these acids, this bicarbonate must be in sufficient amount or sufficient concentration. Otherwise what happens if the value is very low? If to give the ratio 1 means, what happens? All the bicarbonate formed, that will be exhausted within a very short time. So there will not be enough bicarbonate to buffer all the acid load produced. So in order to maintain this pH at 7.4, or in order to buffer all the acids that is produced, the bicarbonate buffer ratio, or the ratio of the bicarbonate to carbonic acid, must be 20:1 ratio. Then only we can attain the normal pH 7.4. So that's about the bicarbonate buffer, and we can move to the phosphate buffer. Again, that for hemoglobin buffer, all this in the next video. So thank you.